Audible, Mithil?
Name, sir.
Thank you very much, madam. एक थोडा वेळ मित्र आपण लगेच स्टार्ट.
काही प्रॉब्लेम नाही.
पार्टिसिपंट जॉईन होत आहेत आमच्या अजून.
तुला कसं कळणार सगळ्यांनी जॉईन केलं? सगळे हात वर करून सांगतात काय मी आलो.
प्रिया जॉईन झाली का नितीन?
Right. Just a minute. Hi, hello, hello, Mital.
Hello, sir.
Hello, how are you?
Fine, fine. How about you?
Right. First of all, thank you very much for accepting our invitation.
I should say thank you for inviting me
That's
for this
great.
lecture series.
That's great. That's great. Very nice. Very nice. Right. So actually, Priyanka will be actually she will introduce you.
Yeah, sure.
Right? She will start, and then name sir will introduce. So we have given this responsibility to all the students,
Yeah, yeah, it's good.
and actually faculty members.
Yeah.
Right. Okay. Good to see you here.
Yeah, me too.
Okay. Good. Good. Thank you. Name sir, करता सुरू?
Yes, sir.
हो, सुरू करा. 4 मिनिटं आहेत. तोपर्यंत इंट्रोडक्शन होत हे होतील 4:30 होतील.
हा मी स्टार्ट करतो. या जॉईन झाली का? पूर्वी Middle, just a moment.
Sure.
तिला बोलायला सांगते.
Hello, good afternoon, everyone. Am I audible?
Yes.
Dr. Nadab sir, am I audible?
Yes, you are audible, Priyanka. Yes, go ahead.
Good afternoon, everyone. I am Priyanka. I welcome to all respected teachers, esteemed speaker, and dear students. This is the second day of our online lecture series on advances in life science research under UGC Stride Component 1 and Maharashtra of Academy of Sciences Kolhapur chapter, which is organized jointly by Department of Biochemistry and Department of Microbiology, Shivaji University, Kolhapur. One important line I must want to say here: nothing in the life is to be feared, it is only to be understood. Now is the time to understand more so that we may fear less. This line said by Marie Curie, scientist Marie Curie. Without wasting time, I am requesting to Dr. Nadab sir, please give us brief introduction about Dr. Mittal Zado ma'am, who is our today's esteemed speaker. Over to you, sir.
Thank you very much, Priya. I hope I am audible to all. Respected dignitaries, my dear student, research student, teachers, good afternoon, everyone. I warmly welcome you to today's lecture, that is metal recovery by bio-ion flotation using biosurfactants. In ALSR 2022, Advances in Life Science Research under UGC First Component 1 and Maharashtra Academy of Sciences Kolhapur chapter. As the theme of our lecture series, we are actually getting some more information of new era which are actually started within the life science. Right. The topic selected for today's talk is one of such attempt because ion flotation is a separation technology evolving for removal of metal ions from the diluted aqueous solutions. This method has its long history but adapted for the laboratory scale only. That is, scale up of this particular process should be required. In spite of its emergence, that is budding science, it has a large potential, right? In commercial hydrometallurgy and treatment of different type of effluent, whatever I know, I just I just putting here. Much more detail we are getting from Dr. Mittal, of course. To know more about this particular growing microbial biotechnology process, we are very fortunate to have our today's expert speaker. So I warmly welcome again Dr. Mrs. Mittal Zadav Zakankar, research scientist at the Helmholtz Institute of Fiber for Resource Technology, Germany. I think I spell correctly. I warmly welcome you, Dr. Mittal, and I take my responsibility to introduce you towards our audience. It's indeed a great pleasure to, and of course, a proudful moment for me to introduce our today's speaker, Dr. Mrs. Mittal Zadav Zakankar, because she is alumni of Department of Microbiology and glorified our department from her success. Dr. Mittal have completed her graduation from Rajaram College, Kolhapur. After completion of her graduation, she complete her masters and PhD from the Department of Microbiology Shivaji University, Kolhapur in 2011. From 2011 onwards, she had almost 9 to 10 years experience as a postdoc research and scientist from the various national and international laboratories, which includes Chungnam National University of Daejeon, South Korea, postdoc fellow CSIR NCL Pune, postdoc from the Department of Power Mechanic Engineering, National Sing University, Hsinchu, Taiwan. From 2019 to 21, she had experience as a postdoc fellow in the Department of Biotechnology at Helmholtz Institute of Freiburg for the Resource Technology, Germany, as a Marie Curie postdoc fellow. Since from 2021 till date, she is working as a scientist in the same institute, that is same German institute. So far, she have more than 20 international publications with 705 citation score and S12 index, and of course much more to become. Obviously in future she can do it. More than publications or other than publications, she had 3 book chapters on her account and 1 book. Recently she had published 1 book. May have to be incorporate this in this thing. So this is a brief introduction towards our today's speaker, eminent speaker, Dr. Ms. Mital Zadav, which is a renowned researcher, good scholar, good friend of mine and Shailesh, and obviously I have to admit it here, and humble person. And it is my privilege to and satisfying to see Mital on that position. So I thankful again to you, Dr. Mital, for for accepting our today's invitation for delivering a talk in our seminar, and in spite of your busy schedule, thank you once again. Over to you, Priya.
Thank you, Priyanka and Naeem, for such a nice introduction.
Our
I would
request
first
to
of
Dr.
all,
Mittal Zadav ma'am to give us her talk.
yeah,
Yes,
I would like to thank Priyanka and Naeem for such a nice introduction. And before I start, I would like to thank all the teachers and organizers of this lecture series for inviting me, and I'm glad to share my research today. So I have some disturbances in internet. So before sharing my presentation, I'm sorry for the inconvenience, but I will have to stop the video. But I will turn it on again after the presentation. So I will just share the screen. Let me know if it is visible. Is it visible in presentation mode?
Yes, 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 yes.
Okay. So as I already introduced myself, I'm Dr. Mithal Zadav, Sakankar after marriage, and today's talk is about metal recovery by bio-ion flotation using biosurfactant. Before I start to introduce my research topic, I would like to give a short introduction about the institute that I'm working at. So I'm working at Department of Biotechnology, which is a part of Helmholtz Institute of Freiburg for Research Technology. This is an institute which is actually integral part of a large institute, which is Helmholtz Zentrum Dresden-Rosentorf, which is again a part of Helmholtz institutions. So HIF, that is Helmholtz Institute Freiburg for Research Technology, is actually situated in the location Freiburg. And we as a department are actually situated in the main campus of Dresden, which is at ZDR. And Freiburg is around roughly 40 to 50 kilometers from Dresden. So here I belong to the biotechnology department, and our department has various themes which we work on, mostly on resource recovery from various primary and secondary sources. So the themes that we have at biotechnology department include bioleaching, which is extraction of metals from primary or secondary resources using various microorganisms and metabolites. Second is biosorption, where this is more specifically towards the separation of particles by flotation, which is usually supported by biological compounds and modified compounds for attachment of particle surfaces. And the other theme that we work on is bioflotation, where microorganisms or their compounds or biomolecules from the microorganisms are used to recover metals, like in the particle form or the metal ion form, using bioflotation. So my research area is more in the bioflotation area, but it differs a little from bioflotation, and it is more specifically bio-ion flotation. The difference is that the bioflotation is more for the recovery of particle matters like ores and minerals, whereas bio-ion flotation, as Naeem already said, it is more about dissolved metal ions. So now coming towards the today's talk, industrial wastewaters are secondary sources of many critical and base metals. What exactly are critical metals is that European Union publishes the list of critical raw materials, which are usually at high risk of supply shortage, as well as have higher impact on European economy, because they don't have it in ample supply. They will have to import those materials. So recent list from 2020 has listed around 30 critical metals, which are at risk of supply shortage, and most of them are metals. And gallium, which is the target metal for this research, is also one of the critical metal. So, recovery of such critical metals from waste streams helps in reducing helps in resource recycling and also reduces the environmental burden. However, recovery of such a recovery is challenging due to low concentration of target metals and high concentration of other unwanted metals in the industrial wastewaters. As you know, it is a very complex nature of the industrial wastewaters, which not only has different metal ions, but also different organics, plastics, and unwanted pH and those things. So it makes the process more challenging. So ion flotation is already being applied for various applications like wastewater treatment, mineral beneficiation, and hydrometallurgy. And it is a promising separation process for resource recycling of metals from secondary sources like industrial wastewaters. Because of its high efficiency to remove the dilute metals and possibility to selectively recover the metals by optimizing the collector and flotation conditions. However, low selectivity amongst ionic species and secondary pollution caused by the use chemicals limits its practical application so far. So, there is a need to develop new ion collectors which are highly selective, efficient, and eco-friendly. To overcome this hurdle, we propose the use of biomolecules as an attractive and environmental friendly alternative. Biosurfactants are surface active molecules produced by microorganisms and have both hydrophilic and hydrophobic moieties. Important characteristics like high surface and interfacial activity, activity at extreme conditions, stability during use, and biodegradability after use, low toxicity, and structural diversity make them stand apart from their counterparts. Rhamnolipid is one such biosurfactant, which is a glycolipid type of biosurfactant and is composed of either one or two rhamnose molecules, which are usually attached to beta hydroxy fatty acid chains. Their high surface activity and metal complexation ability makes them molecule of interest for application in ion flotation. Moreover, extensive foaming capacity makes them a good frothing agent, which is required for ion flotation. So, rhamnolipid, when applied in the ion flotation process, plays a dual role of collector and a frother, or more specifically, a foaming agent, as we described in a flotation. Now, coming to their application in a bio-ion flotation process, these biosurfactants or biomolecules, when we apply in a flotation process, they act as ion collectors and they form the complex with specific metal ionic species. And these complexes, they attach to the introduced air bubbles and they are buoyed on the surface, forming the foam, which can be recovered from the flotation cell. And then it is possible to recover concentrate the recovered metals. So, in order to improve their application in this process, a comprehensive and systematic investigation on their performance and behavior as an ion collector is required. Our objective in this study is to investigate the bio-ion flotation of gallium as our target metal using rhamnolipid as an ion collector and provide an insight into surface activity, foamability, foam characterization for rhamnolipid, as well as influence of these metal ions on various properties of rhamnolipid, their correlation, and finally the flotation recovery of gallium. So this study was carried out in a top-down approach as depicted in this flow chart. During the first flotation experiments, it was observed that mixing of rhamnolipid and gallium resulted in a colloidal solution. Further, it was seen that the collection of concentrates during flotation was difficult due to the high foam stability of rhamnolipid. So we added 1,2-decane-diol, which is a common non-ionic frother, and it imparted a little stability to this foam and also eased the collection of flotation concentrates. In both the cases, it was observed that the foam structure kept on changing with the increased number of concentrates and the bubble size increased with time. These observations altered the course of our study, and it was decided to investigate the influence of gallium as well as 1,2-decane-diol on various properties of rhamnolipid like aggregate size, surface activity, and foam characteristics. So coming towards the aggregation studies, as I said, mixing of rhamnolipid and gallium resulted in the colloidal suspension. So these colloidal interactions were studied using DLS, that is dynamic light scattering. Aggregation behavior of rhamnolipid alone and in presence and absence of gallium and or 1,2-decane diol was studied as a function of pH. Increase in aggregate size, as can be seen from this figure, indicated that interaction of rhamnolipid with either or both of them boosted the aggregation process, again, which may affect the adsorption kinetics and foaming ability of rhamnolipid, which we are not sure, and we can see in the later studies what it revealed. So the main study again was the surface tension studies. It was studied using a bubble pressure tensiometer, which is an instrument to measure the surface tension, and it measures the dynamic surface tension in relation to the surface age. That is, you can see here, the air bubbles are generated at the tip of the capillary in the liquid, and the surface tension of the solution is calculated from the maximum bubble pressure required to form the new surfaces. So, lower the surface tension, less is the energy required to create new bubbles or surfaces, and in our case, new froth or new foam. Studying dynamic surface tension activity is required because it is used to quantify the adsorption kinetics of surfactant and is considered to have a strong influence on foaming properties. The faster the dynamic surface tension drops, faster the surfactant molecules or the complexes of surfactant and metal adsorb at the air-water interface, which increases the foam ability as well as foam stability of the frother. Here in the graph, we can see that the surface tension behavior of rhamnolipid in presence of different concentrations of gallium. We can see here that surface tension curve for rhamnolipid gallium solutions are much different as compared to the rhamnolipid alone, suggesting that the gallium metal has some effect on the surface activity of rhamnolipid. As can be seen, at low concentration of gallium, which is 0.01 millimolar, just a minute, can you see my pointer? Naeem?
Yes.
Can you see my pointer because I'm pointing at the
Yes,
graph?
ma'am. It is.
Okay, yeah. So at low gallium concentration of 0.01 molar, we can see the initial trend of surface tension reduction is very much similar to rhamnolipid alone, indicating that more of free rhamnolipid is present in the solution and it is again predominant at the surface. Whereas in the later stage, we can see it differs from the rhamnolipid alone, suggesting that the rhamnolipid gallium complexes that are formed, they take some time to reach the air-water interface. Again, we can see that as the gallium concentration increases to 0.1 millimolar or 1 millimolar, we can see the complete shifting of the surface activity curve of rhamnolipid towards right. This again suggests that the more rhamnolipid gallium complexes are present in the solution now, and they take more time to reach the air-water interface. And this surface activity can again be explained very well with the help of an empirical relation, which is derived usually from Rosen equation, which can be seen here. And it helps us to derivate the values for meso-equilibrium surface tension, which is gamma m, and T star as a function of gallium concentration. The value of this T star directly correlates with the time required for molecules or complexes to reach the air-water interface, or we can, in other words, we can call it as a diffusion coefficient. Hence, higher the value of T star, longer is the time required for the molecules to reach the interface, and thus higher is the diffusion coefficient. So here we can see that increasing concentration of gallium results in the increasing diffusion coefficient of the complexes. Now looking at the influence of both gallium and 1,2-decane diol, we can see that again, if we add just 1,2-decane diol, it has no such effect as can be seen with the gallium. But when both gallium and 1,2-decane diol are present, again, it shifts the surface activity curve towards right. Again, explaining these results in terms of gamma value and T star value, we can see that the diffusion coefficient for rhamnolipid alone, all with 1,2-decane diol, are nearly same. Whereas those with gallium alone or with gallium and 1,2-decanediol, the T star values are higher, suggesting that the complexes are taking more time to reach the interface. So this was all related to surface activity or aggregation. And now moving towards the foam analysis, which is an integral part of the flotation, as the flotation process is all dependent on the foam ability and foam stability of the frother. So various characteristics like of foam, like bubble size, bubble shape, and water content, have strong impact on flotation process in terms of recovery and upgrading, as it directly affects the foam stability. Studying such properties can provide a valuable perception into the process fundamentals. So we decided to go deeper into these studies. Just to let you know that in the flotation process, produced foam needs to be stable as well as unstable depending on the stage of flotation. The stability of foam plays an important role during the bubbling stage, where the unstable foam will lead to the collapsing of the foam. So if the foam is collapsed, even though the complex of rhamnolipid and metal ion is attached to the bubble, it will not be lifted up to form the foam, and it will fall back in the solution, and so we will not be able to recover this foam or metal and get the concentrated metals. However, in the later stage, as I told you earlier, if the foam is too stable, it does not allow the easy recovery of foam as well as the easy recovery of metal ions. So the foam needs to be stable as well as unstable depending on the stage of flotation. And again, as said earlier, the dynamic surface tension affects the foamability. So I would like to tell you how it affects in that way. So we saw that it affects the formation of bubbles when we measure the surface tension. So here again, it affects the generation of the bubble at the sparger by changing the average bubble diameter, which in turn affect various foam properties. So we did the foam analysis experiments using dynamic foam analyzer instrument, where the foam is generated by in a glass cylindrical column, which is having a diameter of around 40 millimeter, and the foam is generated by sparging the air through a frit, which is usually a P3 glass frit, which is placed at the bottom of this glass cylinder, and air is sparged through it at the rate of 0.2 liter per minute. And we foam this solution for around 50 seconds, and we measure the stability of this foam for around 5 minutes. You can see there's a camera behind here, which is used to record the foaming process, and few images from these videos were selected at specific intervals and analyzed with ImageJ software to obtain the information about various properties of foam, like size, shape, and water content. So as we did this foam analysis experiments for all the rhamnolipid solutions, like rhamnolipid alone, only with gallium, or both gallium and decanediol, or only with decanediol. So now I would like to show you the results of this foam analysis. So these first graphs are shows the influence on the bubble size, and on the left side you can see this graph represents the bubble sizes for rhamnolipid alone and rhamnolipid with decanediol, whereas on the right side it shows the influence of gallium as well as decanediol at various pH. So here we can see that the small bubbles are formed without gallium as compared to the bubble size, which keeps on increasing in the presence of gallium as well as both gallium and decanediol. And these findings further relate very well with the next experiment, that is bubble shape and water content. Now coming to the bubble shape varied from circular during the foaming or at the end of foaming phase to and changed to polyhedral or hexagonal at the end of stability measurements. Here we can see that the circularity of the samples without gallium for rhamnolipid, even if it's alone or if it's with decanediol, the circularity is in the range of around 0.94 to 0.99, as compared to those with gallium, which range from 0.96 to 0.91. In case of the circularity value, the value which is towards the 1, it indicates that it is a perfect circle, whereas the lower value indicates that it is an increasingly elongated polygon and not just a perfect circle. As said earlier, water content of the foam matters a lot in the flotation, and we were able to calculate the water content from this foam image analysis of the foam. Here we can see that water content of rhamnolipid foam without gallium, even in absence or presence of decanediol, it kept on increasing over time, suggesting that the water drainage from this foam is very less. This might be probably due to free rhamnolipid molecules which are available and they make the foam more stable. Whereas the water content decreases in case of gallium and alone or with 1,2-decanediol. In some cases, it increases first and then decreases, but it decreases in the later stage. So here we can say that presence of both gallium or gallium and decanediol, it results in faster drainage of foam, and that's why the water content is low. And we can relate these findings very well with the flotation results that I told you earlier. And we can see already this from the pictures. And as I said, the foams are constantly changing systems. Here, the upper rows like A, B, C are for different pH, pH 6, 7, and 8. The upper rows are for the pictures taken at the end of foaming stage, whereas the lowest like A dash, B dash, and C dash are for the pictures that were towards the end of evaluation phase. So here we can see that the foams with rhamnolipid and decanediol are very fine. They have very fine bubbles and very spherical, and they remain spherical even at the end of evaluation phase. Whereas in case of gallium and both gallium and 1,2-decanediol, we can see that they are circular at the initial foaming phase, but at the end of foaming phase, here we can see that the foam starts coarsening and there's a thinning of bubble lamellae, and it turns dry as the time passes. Also, the shape of bubble can be seen varied, where like it looks circular at the end of foaming phase, where it looks like more polyhedral or hexagonal at the end of evaluation phase. And this suggests the role of gallium as well as 1,2-decanediol in the foam coarsening. Again, if you see the foam towards left side, they look more wet because of the high liquid content as compared to foam towards left, which look more dry because the water drained faster from these foams. And now coming towards the flotation studies. So this is our flotation setup. For the initial experiments, I used to do the flotation studies on the dynamic foam analyzer instrument that I showed you earlier. But this is the flotation setup, which was developed later. This is the picture where you can see this first one is a rotameter, which is used to control the airflow rate, and we can change it as we want. The second is a connection to the compressed air so that we can pass it through the column. This is the flotation column, and here you can see the fourth is a white glass frit, which has a pore size of around 16 to 40 micrometers, and the air is flowed through this. So once you start the flotation, you can see the bubble forming here and the foam increasing. So here is the video of the flotation experiment. Here you can see as the air increases, the foam is formed and it starts coming out of the flotation column. And we usually collect this foam in the evaporating dishes, and we we change the evaporating dish every minute so that we have the different number of concentrates, and we can calculate the kinetics of flotation from this different concentrates that are collected. Once this foam is collected in this evaporating dish, we break the foam and collect it in the small tubes. We measure the volume of this liquid, and then we send it for the ICP-MS to know the metal ion concentration. And from this, these are some results from first few flotation tests which I did earlier. And here we can see that the flotation tests were done at different pH in absence and presence of gallium and decanediol. Now here in the table, you can see that the highest recovery of gallium can be seen at pH 7, but here there is another value in the table which is called as upgrading value. And we can see here that the upgrading value is higher at pH 6 and not at pH 7, even though a recovery value is little less. This upgrading factor is important parameter when considering the flotation recovery in presence of other metals, like when we are talking about industrial wastewaters or module solutions with different metals. So for the flotation process with metal mixture, higher the upgrading value for the target metal as compared to other metals, higher is the selectivity and process efficiency. So we would go for the optimizing parameters where the upgrading value is more as compared to recovery, because anyhow if the upgrading is more, you will selectively recover your target metal in higher amount. I have not included the recent results here where we did the flotation experiments of gallium for the selective recovery in presence of other metals like arsenic, because we want to apply this flotation process for recovery of gallium from gallium arsenic vapor industry, which has more amount of arsenic as compared to gallium. And our recent experiment has showed us that our experiments allow us to recover gallium at higher concentration very selectively as compared to arsenic. So coming to the conclusion, I can say that we were able to show that various secondary responses of flotation like water recovery or bubble size and bubble shape, they are all reflected in the principal responses of flotation like flotation grade or upgrading and recovery of metals. And we already saw that these gallium and 1,2-decane diol has very influencing effect at basic fundamental properties of this frother, and it affects a lot. And adequate control of these parameters could provide us additional benefits for foam flotation with respect to recovery and grading of our metal ions. And these experiments show that gallium recovery was highest at pH 7, but the upgrading value was more at pH 6. So thank you for your attention, and you can ask me if you have some questions. I
Hello.
hope
How
you
are
were
you?
able to understand because the flotation is new and it has more physical and processing terms and interactions as compared to more biology.
Awesome. Thank you, ma'am. It was really a great and informative lecture for all of us. As we all know how biosurfactants are important with respect to their applications. And here you have explained most important points about the biosurfactant and its flotation studies. Definitely, it will helpful for all the student in their future research activities. Thank you, ma'am. Thank you so much.
Just a moment. Here we have lots of audience, Mithil. So I appeal participant, mostly the girl participants who required to go for the higher studies can ask some question, or if in that way anyone will having any kind of question, they can really ask me. Is it okay with you?
Yes, yes, yes. You should ask freely, and जर तुम्ही मराठीमध्ये जास्त कम्फर्टेबल असाल तर इट्स फाईन, तुम्ही मराठीमध्ये विचारू शकता.
So you have a platform student, you can take advantage of it. तुम्ही विचारू शकताय काय मॅडम आहेत इथं, काही शंका वगैरे असतील.
आहे ना तुम्ही सगळे?
सगळे आहेत प्रेझेंट, पण कुणाला काहीच प्रश्न
पण
नाहीत,
कोणी
कारण
विचारत
तू
नाहीये.
लेक्चर एवढं व्यवस्थित घेतलायस की त्यांना काहीच प्रश्न नाहीत.
किंवा मग काहीच कळालं नाही. आयदर दिस ऑर दॅट.
वी विल टेक इट पॉझिटिव्हली. Anyone, guys, you have any question? If you want to ask any opportunities in abroad, if you are anybody
तुम्ही
interested.
काहीही विचारू शकता, नॉट ओनली जस्ट विथ द रिसर्च पॉइंट ऑफ व्ह्यू.
This student, ma'am, is alumni of our department. She has traveled from Shivaji University to South Korea, and then from South Korea to many other countries. So if you want to go outside, what are the hurdles? You can may ask to ma'am.
आपणच
Yes.
लँग्वेज ऑफिशियली मराठी करू म्हणजे कदाचित ते विचारतील.
Hmm.
उलट मुलींना तरी जास्त प्रश्न असले पाहिजेत. आम्हाला खूप होते. मुलींना नाही तर मुलांनी विचारा, की तुम्ही कसं सपोर्ट करू शकताय मुलींना, तुमच्या बहिणीला, तुमच्या होणाऱ्या बायकोला, व्हॉटएव्हर किंवा तुमच्या ओळखीतल्या मुलींना. म्हणजे ॲटलीस्ट तुम्ही विचारलेल्या क्वेशन्सनी त्यांना त्यांचे आन्सर्स मिळतील.
Yes, students.
कदाचित सेपरेट लेक्चर घ्यायला लागतं त्याच्यावर. आत्ता नाही आहेत क्वेशन्स तर.
Okay, thank you, ma'am. Thank you so much for giving us a very great and informative lecture. Thank you, ma'am. Now I'm requesting to Mr. Nitin Naik, sir, for vote of thanks.
Thank you, Priya. This online lecture series organized jointly by Department of Biochemistry and Department of Microbiology under UGC Stride Component 1 and Maharashtra Academy of Science Kolhapur chapter. For this lecture series, we got permission from higher authorities of Shivaji University Kolhapur. I am very much thankful to all the higher authorities, Honorable Vice Chancellor Sir, Honorable Pro Vice Chancellor Sir, Registrar Sir. Again, I am thankful Head Department of Biochemistry Jadoo Madam, as well as Head in Charge Department of Microbiology, Professor K.D. Sonone Sir, for organizing this very nice lecture series. I'm also thankful the delegates from India as well as from abroad for attending this lecture series. And I'm very much thankful Dr. Mittal Jadoo Madam for accepting the invitation from both departments and delivering such a nice talk on metal recovery by using bio-ion flotation using biosurfactants. I'm very much thankful to all the faculty members from Department of Microbiology, Biotechnology, and Biochemistry. I'm very much thankful to all the students from all the streams of HM, that is Microbiology, Biochemistry, Biotechnology, MSc Medical Information Management, PG Diploma Bioinformatics, and other. There are few students from the colleges which are affiliated with Shivaji University have been also joined for this lecture series. I'm happy to share with you, ma'am, there are more than 250 registrations are there for this lecture series. And it is a continuous lecture series. Still, many more lectures to be conducted under this lecture series. At the end, I'm thankful to all the participants, delegates, speaker, and all the higher authorities. And with this, I am very much thankful to one and all who have joined for this lecture. Thank you very much, ma'am, and thank you all. Over to you, Priya.
Yeah, thank you.
Thank you, Nitin, sir. Shall I proceed, sir, for to you? Okay. Hello, sir. Am I audible?
Yes, yes.
Okay, sir. Can I proceed for to leave with the permission?
Go ahead, Priya.
Okay, sir. Thank you, sir. Okay. Thank you, everyone. Thank you.
Thank you, one and all. Thank you, Nikhil, madam.